Welcome everyone to the second CCS Lounge community event focused on the science of COVID-19 and its impact on UCSB teaching, research, and campus life. My name is Gerardo Aldana, and I have the privilege of serving as the Dean of the College of Creative Studies. CCS is that small college campus experience nestled within the resources of a Research One university. With the CCS Lounge, we aim to simulate the kinds of informal conversations that occur in the social spaces of our funky little building. Meaty content, relevant topics, casual conversation. This is not meant to be any kind of formal briefing, but a discussion amongst engaged colleagues. For this, our second CCS Lounge, the college is delighted to be co-hosting the event with UCSB alumni. Now, before turning to our moderator and our panelists, we'd like to share with you a special message from UCSB Chancellor Henry T.
Good afternoon, and welcome to our College of Creative Studies launch event, hosted by CCS and the UC Santa Barbara alumni. As we continue to face the many challenges related to COVID-19, we are just delighted that you are joining us for a conversation today on how the pandemic has impacted our university's teaching, research, and especially student life. Many thanks to Dean Aldana for his leadership of CCS and his vision in bringing about the discussion for our community. And thanks to our faculty panelists, Professor Carolina Arias, Stuart Feinstein, and Armand Curious, for sharing their expertise with us. Our campus has also been fortunate to benefit from the research and the leadership of Professor Arias and Feinstein as members of our COVID-19 working group, which has been meeting nearly daily since March. Professor Feinstein serves as our COVID-19 response team coordinator, and Professor Arias has worked tirelessly to help develop a faster, less expensive, CRISPR-based COVID-19 test. Professor Curious is a recently elected fellow of the American Association for the Advancement of Science. Congratulations. He was honored for his important investigations of the ecological role of infectious agents in ecosystems. Now, a special warm welcome to today's moderator, our distinguished alumna, Angela Belcher. Angela graduated with a Bachelor's of Science from CCS in 1991 and a PhD in chemistry in 1997. Dr. Belcher is the James Mason Crafts Professor of Biological Engineering and Material Science and Engineering, and the head of the Department of Biological Engineering at MIT. I fondly recall welcoming her back to our campus as our CCS commencement speaker in 2003 and our graduate division commencement speaker in 2006. In 2017, she returned to celebrate the 50th anniversary of CCS with us. During that time, she helped tremendously with fundraising and events and advocacy for our wonderful CCS. Angela, we are always so inspired by your presence, and we look forward to the day when we can welcome you and all of our community members back to campus when it is safe to do so, hopefully very soon. I will now turn the program back to Dean Aldana, who will provide more details on this event. Thank you.
Thank you, Chancellor Yang. Just a couple of final points before we get started. The overall event, it'll last until 5:30. It's going to be a moderated conversation. It'll be informative, yet fun and lively. We do encourage you to submit questions through our chat or the Q&A box, as we've allocated a little bit of time at the end for your question. And so with that, I'll add my quick thanks to the panel and hand it off to our moderator, Professor Angela Belcher.
Thank you, Dean Aldana. It's a pleasure to be here with this very distinguished panel and to be back almost in the College of Creative Studies Lounge for the kinds of great intellectual discussions that I have fond memories from my time as a CCS student. So as my panel gets ready, this is going to be a science discussion, mostly about the science of COVID and SARS-CoV-2. And we're also going to talk about its impact on teaching and what it means for the future biologists and biological engineers who will one day step up and make the newest advances in the field. So I first wanted to talk to Carolina about her work. Some of the things that my fondest memories to this day about being on campus at UCSB is it's the most interdisciplinary university I've ever been to or visited. And one of the things that has always been amazing is how faculty and students from around the campus come together to solve problems. And this is, one of the most challenging problems of our lifetime, this pandemic. So can you talk a little bit about your experience in this interdisciplinary or transdisciplinary research, and also the very fascinating area that you work on in viral detection, why it's, how the interdisciplinary part came to play in that, and some of the things that you found most interesting and rewarding about it?
Oh, absolutely. And yes, Angie, it's been really impressive to see how our colleagues from so many different disciplines have come together to respond in this time of crisis. I think one of the best examples that I have is a series of talks open to the public that were put together by Ambush Singh and Joseph Water from the Center of Information Technology and Society, where they brought experts from so many different fields, including some people in infectious diseases, from economy, from physics, talking about very crucial questions that have helped us understand and cope a little bit better with this pandemic, from how does climate alter or influence transmission of pathogens, what are the impacts, economic and social, and the disparities of this pandemic, and of course, basic information about infectious diseases. But as you say, I think one of at least for me and from my personal experience, one of the best examples that I have of interdisciplinary teamwork was when we came together with 3 faculty from my department: Max Wilson, who is a molecular biologist, Kane Kosick, who is a neurobiologist, Diego Acosta, who is a cell biologist, and myself, who I am a virologist. I must say, I did not work with coronaviruses before this pandemic. I work on herpes viruses and Zika, asking completely unrelated questions. But I clearly remember in early March, when we saw that this pandemic was reaching the proportions that we hadn't seen before, we had the need of doing something for our community. And I remember talking with Max, Ken, and Diego, and they shared that emergency of we have to respond somehow. So we decided to come together and establish here at UCSB all the tools that we needed for us to be doing testing. At that point, if you remember in March, it was impossible to get a test for COVID. You had to be in the hospital for you to be tested. But we saw that was a very important aspect of pandemic control, right? You need to test people to see who has it so that you can mitigate the outbreaks. So I remember we sat down and we talked about this, and that same day we just ordered all the reagents that we thought we were going to need to establish the testing here. And within some weeks we were able to do it. But at the same time, we saw that everything that we needed, all the reagents and equipment that we needed to do testing, were in so high demand that they were almost impossible to get. So at that point, we came together again. This was when we still could actually, get together and people could talk to each other in not soon. And we said, okay, we need to do something else. So we decided to develop a new testing method that was independent of the reagents that were used at that point, which if some of you may be familiar, one of the most widespread methods used is RT-qPCR. And at one point we couldn't even get the machines to do that test. So we decided to explore other avenues, and we used, as the Chancellor said, a method that relies on CRISPR. Many of you probably have heard of CRISPR for genome editing. And we use a different variant of it, and you can think of it as molecular scissors. So these molecular scissors, when they are in the presence of the viral genome in a sample, they kind of go crazy and they start cutting everything. So we use that to cut a reporter that has fluorescence. So when the reporter is intact, it doesn't fluoresce. When it's cut, it fluoresces. So when the scissors get active and get crazy and they release the fluorescence, we have a sample that is going to fluoresce, and that sample is going to be positive. And we can detect that with very simple machinery and not specialized equipment. So it's been very useful. And we use that method to do surveillance here in our campus of about 2,000 students and faculty that were asymptomatic. So this was in May when we were starting to understand that asymptomatic individuals could actually transmit the disease, and we decided to focus on what was going on here in our campus. And so we detected some asymptomatic cases in our students, and that actually represented the leading edge of one of the outbreaks that we saw happening in June, July. So it's been. I also have to mention that for us to be able to do that, we established a lot of different collaborations with our Santa Barbara Cottage Hospital, with public health, with student health here. We had support from the Office of Research. So again, it's this sense of our community. Everybody came together and we started working on this. And I think we really made a lot of progress and solidified the basis of the testing program that we have now on campus, which is pretty successful.
So that's pretty amazing. Were there any students involved in that? I just imagine being a young biologist or a young person getting to work on such cutting edge technology that you're actually then testing it within a couple of months on campus and getting real data, real world data. Were students involved in this?
Yeah, so we did have our grad students involved in this. We actually have CCS Alumni who was involved in this effort. Unfortunately, when we started working on this project, our undergrads could not come back to the lab, so we didn't have undergrads working actively in this particular project. But I must do a shout out to many of our CCS undergrads that are in our labs that have worked remotely, helping us curate a lot of the information and a lot of the manuscripts that come out, that is by the hundreds, if not the thousands daily. So even when they were not here pipetting directly, they were still involved in the research, and they have been absolutely essential and valuable for our endeavors here.
That's great. For about 10 years, when I give talks and lectures, I always say there's never been a better time to be a scientist or engineer than now, because there's never been a time when we have the possibility to make such a big impact. And that can be in medicine, it can be in energy, it could be in the environment, so many different topics. But it has been really great to see how involved the undergraduate students are and the graduate students are in this very important moment in time. So thank you for sharing that question. So we all know that one of the initial key components of getting a handle on this disease and this pandemic is testing. And so Carolina gave that example of, she didn't work in testing. She and her colleagues came together to push the limits of the technology so that you wouldn't have to use very expensive equipment to be able to do this. But the other really important aspect of this is vaccine development. And I'm going to invite Stu to come in and give us some of the science behind that. So as many of us get our flu shot every year and really hope it works, or we know about many diseases where vaccines are being developed but haven't been successful. Can you talk a little bit and give us an idea about how this one so rapidly developed in under a year to something that looks like it's pretty effective?
Thanks, Angie. That's a really good question. And I think you and Carolina just set up how I would answer that by, you were both talking about all the tools that it's such an exciting time because we've got all these great tools. And I think to understand how this vaccine works so fast, you have to look at history. And it's a history of 40 to 50 years of major investment into basic research by the NIH and many other organizations, asking basic questions in molecular and cell biology. So how do cells work? What are the molecules involved in? How do they work? What can they do? And those decades of research provided us with two things. It provided us with an intimate knowledge of how our cells work and how our immune system works. And so that then we can use them, we can manipulate them to do things that we want to do. And the second thing that's incredibly important that all that basic research did was it provided us with an arsenal of molecular tools, things like DNA polymerase, reverse transcriptase, all these things that we can mix and match to do new things, to do creative things, to do essential things. So in a way, it's analogous almost to the computer science industry, where you've got all this computer hardware and you've got all this great software available to you, and you're kind of limited by how clever can you mix and match parts to do new and interesting things. And that's really where molecular biology is. And that's because of all the great work that was really done starting back in the 70s and the 80s. So how does that impact, get back to your question, how does that impact on a vaccine? So just a little bit of immunology. Our immune system is very good at telling self from not self. And it's kind of brutal about it. If it identifies something as not self, it makes antibodies to destroy it. So that's at the heart of a vaccine. The traditional way to make a vaccine would be to take a disrupted virus, say a polio virus, make a bunch of polio virus, treat it really roughly, disrupt it, and then inject that into people. The people's immune system would recognize, I've never seen this polio virus debris, this broken stuff, so it's not me, I'm going to destroy it. And so that's the traditional way to do it. More recently, using many of these tools that were discovered with basic research, we could, instead of injecting a broken up virus into somebody, we just use recombinant DNA to make a whole bunch of one of the viral proteins and inject that. That has a big advantage of it's by definition much safer because there's no virus there. There's no infectious agent even broken up. It's just the protein. But that is still time consuming and labor intensive. But that's the strategy that's being used for some of the new vaccines that are out, not the mRNA vaccine that I'll finish up with in just one second. But you can use recombinant DNA, for example, to make a whole load of COVID-19 spike protein and inject that into people, and you will get an immune response. We know that works. The mRNA has a couple of really interesting advantages. Instead of introducing the spike protein, for example, directly into a person, we introduce the messenger RNA that has the information to make the spike protein. So you inject the messenger RNA, it goes into the cells, our own cells, and it tricks our own cells into making the virus spike protein, which it then releases to the extracellular space. And now there's a foreign protein there. Your immune system recognizes it, and it makes a whole lot of antibodies. So now if subsequently you're exposed to a COVID-19 virus, you've got antibody sentries ready to disrupt it before it can make you sick. That's the principle. The advantages of the messenger RNA, and these are the first time messenger RNA viruses have been used in people, are a couple fold. First, you introduce it, it instructs the cells to make the protein, and then it's gone. Second, it's easy and quick to make. Much easier and much quicker to make than using recombinant DNA to generate a protein. And most importantly, and I think maybe some of the questions later may get to the new variants that have sprung up in the system, but the messenger RNA approach can pivot on a relative dime. So some of the companies are already talking about if any of these variants turn out to be problematic for our current vaccine, they can retool the messenger RNA vaccine system very quickly. And now we'd have a new vaccine that could see the variant as well as the original one. So it really all started with this investment that was really quite remarkable, that especially the NIH in the United States made this huge investment in basic science. And we're now reaping, this is only one example of reaping the benefits of that enlightened approach to funding science.
Well, thank you, Stu, for explaining that. I think it's great to, so all four of us live deeply in the world of thinking about antigens and proteins and DNA and mRNA, and it's been really impressive to me how these terms have really come forward as being part of everyday words and conversations. But when you first start thinking about mRNA, I remember from beginning biochemistry, you're taught that RNA is not as stable. And so I think one thing that's really fascinating about the technology that developed around the ability to deliver a message are these nano carriers that help stabilize it and deliver it where it gets to go. And so I think that's such a neat example of the cross-disciplinary, transdisciplinary research where okay, now you have to figure out how to package it to get it in. And so I think it's all very very exciting. Were you surprised that it works as well as it does so far?
I was very surprised, and I'm not an immunologist by training, but some of my friends who are, they were hoping for something to be 60 or 70% effective, not 94 and 95% effective. And you raise an interesting point, Angie. Just everybody knows that there's these extreme cold temperatures are necessary for the vaccines. And that's partly because the way the messenger RNA is not just injected naked into a person, it's packaged in these fatty lipid nanoparticles. And that's necessary to get the messenger RNA to actually enter into the cell. But the composition of that falls apart quickly at warmer temperatures. And that's the reason for the cold that has been so much a part of the conversation.
Thank you. So we've talked a little bit about testing and then development of the vaccines. Armin, can I bring you in to talk about transmission and around preventing transmission?
Sure. Thank you, Angela. So the way I look at this as a disease ecologist is that for any pathogen, there are two issues and they are in sequence. So you never get a disease without encounter. And encounter has two components: the mixing of space, the host and the pathogen have to be in the same place, and there has to be a set of behaviors that permits the pathogen to enter the host. And then following that, there are compatibility issues. The pathogen must be able to develop in the host, acquire nutrition from the host, and reproduce in the host, and also avoid or mitigate the host defenses. So looking at, and these are in sequence. Nothing happens in the host without the encounter issue. So transmission to me is always a place where the biggest impact can be made most quickly, because if you block transmission, the problem has been resolved. So here, of course, that has not happened with COVID. And so the numbers game is about reducing the total number of viruses out there, and in particular, those engaged in transmission that have somehow left the host to, I can't say seek because they are passive particles, but to encounter the next host. The solutions for that are now well known. Masks reduce particles coming out of the host. Even pretty crummy masks do that. The fancier masks also reduce significantly the likelihood of the viruses entering the host. So it's actually altruistic. That's the main effect of wearing the masks, is reduce the risk to other people. High quality mask protects you too. The second thing is social spacing. This virus has limited mobility. It is in droplets, basically. And so the greater the distance between people, the less transmission. And then finally, quarantining, separating infected hosts from the uninfected hosts. Those are the the tools in the transmission game. And of course, as we now know, they also, all of those things exist in a social, economic, and political environment. So this is not simply the kind of thing you can just model in a computer without taking in all those considerations. I'll just offer this as a sort of a little mental game. We could call it a little model. One of the most important features of the virus is in hosts is it appears to be transmissible for a relatively brief period of time, something in the vicinity of 10 days. So what that means, if you were to shut down a society for, lockdown for, let's say, 3 weeks, as New Zealand did with its now famously very small initial problem and quick response, they took it to 0 or near 0, and near 0 means you can follow up on things and contact and deal with the very few people who have it. Now, here's what's interesting. Let's say half the people in the country are infected. A disaster. Shutting it down for 3 weeks takes you to essentially the same, near 0. The last thing I think that is very pertinent, and obviously those things only happen with a social and political and economic milieu where those things can be done. And societies across the world have, and even across states, have varied tremendously in their willingness and ability to do that. The last thing I want to just talk about is the relatively new news and what this means about transmission for these variants that have been detected in the United Kingdom, in South Africa, Brazil, and now in California as well. First thing to be said about that is mutations happen presumably at random. You get more variants when you have a lot more virus. So you're going to see this event, which is a mutation isn't evolution, it's just like the kickoff for evolution. Without the variants, you can't alter gene frequencies. So these tend to happen in places where transmission is rampant, where it's happening the most. You get the most virus there and the most opportunity for variants. And the last thing I'll say about this, and my concern for dealing with this, is that if a variant is spreading, and apparently, for instance, the best studied one is in the United Kingdom, it has spread rapidly, it suggests that it's under positive selection. And we now do know that this is more easily transmitted. And it's no surprise that it has shown up in Brazil and in California as well, different variants. The last part about this is if it's easier to transmit, that opens up selection for further ease that can build on the mutations. And it also opens up selection for greater pathogenicity. And this is, of course, what we are all really fearing, is that as this goes on, the virus becomes more pathogenic, causes more morbidity, and in the worst case, more mortality. The broad theory about disease transmission is that the easier a pathogen is to be transmitted, the more it can be selected to be more virulent, because it doesn't have to wait for time for hosts to slowly release it. So that's my perspective on the issue of transmission and looking at this from the eyes of a disease ecologist.
So thank you, and that brings us back. I'm going to bring Carolina and Stu back into this conversation, because as we talk about these variants and as we talk about the mutations, we know there's the mutations at the genetic level that there's at the genetic level, which changes the proteins and how the proteins are going to be interacting with the cells during the infection. So when we come back, we're going to have a poll. Our poll is going to be topics around the virus. And then after that, I want to come back to Stu and Carolina and ask about detection, the different ways of detecting the virus through antigen or PCR. And if we could explain that a little bit, and then explain maybe how we can make modifications for these new variants, or whether we even need to make modifications for the new variants, and how we might be able to tell. So let's start with a poll. Everyone should be able to take this poll. The questions are going to come, I think, one right after the other. So the first one is, how big is the genome, the entire genetic information of SARS-CoV-2? Okay. The next one. Oh, there's the answer. Oh, let's see. Is that the answer, or? Okay, so 30% of, it was pretty evenly split here. The answer is 29,811. So 30% of people got that right. And Carolina said that that's a pretty big size of a genome for a virus, but it's actually quite a bit smaller than humans, which is around 3 billion, or E. coli, which is around 4.6 million. The next poll question: how many coronaviruses, how many known coronaviruses can infect humans? Wow, great job. So the answer is 7. Very great, a lot of information from our audience today. The next question is about the host, the early host. What animal did scientists think that SARS-CoV-2 originally came from? Pigs, horses, bats, or cats? Yep, overwhelmingly people voted bats, which is the correct answer. Well, it's the correct answer where we think it came from. And the next question, if we could show the next poll question: when was the first case of COVID-19 reported in the US? Again, majority of people got it correct, January 21st, 2020. Of course, I don't think we knew it at the time, but we figured that out later. And then the last question is, how many people participated in the Pfizer COVID-19 vaccine clinical trials? That's right, 44,000, and 79% of our viewers got that right. So thank you for participating in that poll. I hope that you enjoyed it. So I wanted to come back, Stu and Carolina, to ways. What are the major ways of testing right now for the virus? And how is that going to be important in looking for variants? Are we going to have to go and sequence the entire genome to understand if it's a variant or whether it's the original coronavirus that we had in the US. So maybe we can compare and contrast current technologies for detection and how that might affect looking for the variants.
Stu, you want to take the tests?
I can start and then you fill in, Carolina. So the testing that's done now is largely, the most common test is a RT-PCR test, which is a polymerase chain reaction type of a test. And its most important characteristic is that it is very, very sensitive. So it can detect a very small number of viral particles. So generally there is a nasal swab or a saliva sample, and it gets subjected to an RT-PCR reaction of one form or other. And that's looking for the virus itself. Okay, and the important part about that is time is everything in testing. So when we first started testing in this country, there was a company called Quest, which had was a big player in the testing early on, but it was often taking them 5, 6, 7, 8 days or more to get the result back. So if you think about transmission the way Armin spoke about it, if it takes 5 or 6 or 7 days from the time you take a test and maybe contagious, maybe infectious, and you don't know for 7 days, you can pass it along to a lot of people in that duration. So time is everything, and we really like now to get test results back in a day or two if we can. There's another kind of a test where you don't look for the viral product, the viral RNA itself, but you look for viral protein. So that would be an antigen test. And there's a lot of those that are also out there. And then there are clever and creative ways. So these can be used, but those won't distinguish what we'll call the original virus from a variant. In order to do that, you have to do what you were mentioning, Angie, and that you have to do sequencing of the virus. You can sequence not necessarily the whole virus, you can just sequence the spike protein, and that's where most people are looking because that's where most of the viruses are. And Carolina and I have been talking about doing this for our UCSB community in a matter of somewhere between days and weeks, we hope. And then, so that's how you have to get the variants. There are some tests, Carolina and some of her colleagues have some very clever ways that she's been talking to me about lately, where we may be able to determine not if it's a brand new variant, but you could say, are any of these 100 people, do any of them have the British variant? Or do any of them have the South African or the Brazilian? And so she has some clever ways that we may be able to do that without sequencing everything in sight. And Carolina, do you want to add anything?
Sure.
Or about the vaccine maybe too?
Yeah, so of course, I mean, we've been bombarded with information about the new variants coming and all of the concerns that having these variants implies, not only for transmission, as Armin was saying, but for detection, right? So some tests can actually miss some of these variants if they are looking in the region where these mutations happen. And we do know that some tests are going to be less accurate, right? And these are only 4 variants that we have detected so far, but the virus keeps mutating. This is what viruses do, right? So there's going to be the accumulation of other mutations. And we may be unlucky enough that some of the tests that we do are going to be invalidated or are going to be not sensitive to those variants. So we need to keep track of what is the population of viruses that are circulating out there. How do they look? What changes do we have? And we can achieve that by doing what Sue was saying, sequencing, right? So as soon as we get positives, we just sequence the entire genome, or paying particular attention to the spike, which is the spike protein, which we have heard of a lot, which is one of the protein that is going to interact with the cell, and it is also going to alter how some of these viruses are transmitted, if there are changes in this protein. That's what I mean. So one of the main concerns that we have right now is what is going to happen with these variants in the vaccine, right? So everybody is in hyper alert, and the companies that have made these vaccines are right now doing a lot of studies on seeing how effective is the immune response elicited by their vaccines to neutralize or to prevent infections or to basically help respond to these infections. So far, the data that has come out shows that some of these vaccines remain active, at least to the level that they will provide protection against these variants. Now, I know that also Pfizer and Moderna, Moderna just reported this, they're going to start developing some vaccines that can be a booster, right, that are going to target these specific variants. And that is one of the advantages that these mRNA vaccines have that some others may not, right? It's like they can be tweaked and changed very quickly. Than if you had to redesign an entire new virus or an entire new particle, which takes longer. So changing RNA is easy, right? It's like we can go and basically just write the sequence of RNA that we want and then put it into these little packages and deliver it as a vaccine. So all of this is proactive work that these companies are doing that are going to help us deal with the variants. But one of the most important things that we have to keep our eye on is what is out there. How are these viruses changing? What mutations are they accumulating? And whenever we start seeing the prevalence change in one variant or the other, we have to be in alert. Is what Armand was saying, right? It's not only the transmission, but are these variants causing different types of disease? Are they affecting now new groups? Are they being more lethal? We don't know, but we have to be on alert.
So thank you. That was really interesting. So when you were talking about the RNA versus making the whole protein that would be the antigen based, so the RNA is being synthesized in the lab, not necessarily by an organism. So it can be changed much quicker and not have to be packaged by an organism, but instead is packaged within these self-assembling organic nanoparticles. So that what you're saying is that allows for much faster changes because you go in and you change it on the computer, and then then you're able to synthesize it based on that.
Exactly, and what the companies think is that you can get probably a booster, right? So even if you get vaccinated with these particular strains that is circulating right now, and then a new one comes, you just get a booster for the next one, right?
Yeah, that sounds definitely promising. So I wanted to just switch a little bit with the time before we take questions from the audience about the impact on students in terms of future careers. I mentioned now we're probably going to see a big increase in students wanting to be biology majors and biological engineering majors and interest around infectious disease. As my own students moved off campus and were doing remote lab work, like Carolina said, her students were doing, computer science also became much more an important part of the equation in terms of prediction, large data sets, and things like that. So one of the things I've encouraged students to do is to increase their computer science skills and coding skills. What are the kinds of advice? What kind of advice are you giving students now? Armand, I know that you're teaching a special class.
Yes, it's a CCS course, a discussion course on COVID epidemiology, focusing on transmission. And as to give it to students is always risky and uncertain because I don't know the future and nobody does. So you have to, I think, offer advice that is precautionary and flexible. So current in the United States, given chances, rushing out into the job market sounds risky to me. So if you can, stay in school. That, of course, has serious economic issues. And also the institution would like to graduate people. So that's something that people should think about. The career that I think is already of increasing interest is public health. So people who are interested in biomedical careers have traditionally, in overwhelming numbers, been focused on the clinical track. And this is everything from physician through nursing, well, all the way down to orderly. They deal with people, their hands are on people, and they offer therapeutics. Public health does not actually deal directly with people, but with much larger numbers of people. And so it's a career for a very different mindset. And these days, seeking a master's in public health sounds like a very interesting option, and one which many students, as soon as they hear about it, are really interested in, because what it does is it gives you a skill set in research design, experimental design, statistical analysis, data management, and is very directly dealing with these issues. So that's a career direction that I think is likely to become much more popular across the nation. And that is probably a good thing.
Thank you. So there, I'm going to pull out some questions from the audience. One of them is a little bit of the first one's a little bit of a policy question. And I'm not sure, Stu is a coordinator on campus, if this has been decided, because this is controlled at the state level, but should parents work on getting their students a COVID immunization, or will UCSB have the campus clinic manage that? Can you update us on what the thoughts are?
I can do that. I mean, the campus is getting ready and has already distributed a small number of vaccines. And the bottom line on vaccines is there are way, way, way more people hoping to get a vaccine than there is vaccine availability. And hopefully that's going to change over the course of the next few weeks and months. However, at this point, the advice that everybody is giving to everybody is, if you have an opportunity to get it anywhere, get it. And as the allocations are sorted out, it's going to be by highest risk factors. So the major risk factor is age. And so that's why now the state of California has approved initially healthcare workers, because they're on the front lines every day with people who we know for a fact are positive. But then it's people over 75, and the governor recently extended that to people over 65. But in Santa Barbara County, for example, there's not enough vaccine to go around to get everybody who's over 75. So that's still the highest priority. And as you move down with age, I'd say most of our college students are going to be not near the top of that list. If there's a way to get them a vaccine somehow, that would be a very good thing to do.
Thank you. So this question, the next question, kind of relates to that, and I'm not sure if Carolina or who wants to take it, but it has to do with we're all familiar with the basically the inactivated whole virus that's used for the antigen vaccine. And originally, when the vaccine was starting to be talked about, I thought that's the one that I want. But when the data comes out, you know, the mRNA one is working so well. So the question is why aren't we just using the old method of making the inactivated virus? Would that speed things up? I guess that's the question.
Carolina, you want to start that one?
Sure. I don't necessarily think that would speed things up, right? Is the development of the mRNA vaccines has been extremely successful and has been very, very quick. Usually, I'm sure most of you know and have heard, it takes years, if not decades, to get a good vaccine or a vaccine, let's say, developed. One of the things that has shortened the time is, again, the capacity of changing these mRNA sequences very quickly, right? And designing the ones that are best. It is a new technology, but all the data that has come out shows how effective and how safe it is. That nobody was jumping the steps in the validation. There was no jumps in the safety testing of the vaccines. The clinical trials were done as they were supposed to. And there were a lot of factors that converged into having this vaccine being tested and validated so quickly. First is we had a lot of people that are being exposed to the virus. So that helped kind of speed up the clinical trials that usually take longer if the disease is not too prevalent in the population. That is not the case with COVID. It's very prevalent and it's out there. So it was easy and fast to get people in these trials. The ones where you have either the inactivated virus or you have other pieces of the virus that are going to be a protein-based vaccine or a peptide-based vaccine, they may take a little bit longer to develop, right? There were some that may not elicit the same kind of response or a good response that is going to be offering the proper protection. So I don't think that we're saying one vaccine is bad or the other one is the best approach. Is in this case, the mRNA vaccine showed to be very good, right? There are other candidates that are being right now tested and developed. It may just take more time to change them. So again, it's not one is bad or the other one is good. Is the science was there and the convergence of all these factors were there to allow this vaccine to move fast, right? And just because it's the new one of this kind doesn't mean that has not been validated and tested for safety as others have been before.
Angie, if I could add one more point about the vaccine, and this, I get asked this question daily. And the question is, don't I worry that the messenger RNA mechanism being a nucleic acid is going to alter my genome, my own genes? And I would ask people to go back and remember their high school or college biology class and to remember the differing role of DNA and RNA in cells. Your genome, your genes, is your DNA. RNA comes later in the process. And your DNA is there forever. RNA is there for a little while and then it gets degraded and tossed out. So there is no way that an mRNA molecule is going to change anybody's genome. So I think that that's a very important, it's not an unreasonable question for people to ask if they have to think back 40 years to their high school biology class and which molecule does which thing. But I think that that's something we can say with complete assuredness that it's not going to affect anybody's genome.
I think there's a compartmentalization issue as well,
Right.
That they're not localized in the same place.
That's right.
And one of the things that makes RNA, unstable in this case is a big advantage because it's unstable, it doesn't stick around very long.
Right.
Let's see. There was a question about the new normal. Let's see. Basically, I'm not sure exactly where that question went, but it was about what does it mean getting a handle on COVID? What does that look like? Are we going to be wearing masks for many years? Are we waiting for herd immunity? What can we say about the new normal?
Yeah, that's a question. Armand, do you want to?
Yeah, that's of course nobody knows is the most honest answer, but I would project that at some point, even in this very mixed up and fairly sloppy world, that the virus will become uncommon enough that it can be managed by reduced transmission. And also the one area where there's been already, I mean, more lives have been saved probably by therapeutics than anything else. And the ability to treat people with this will continue to get stronger and stronger. Everything from just the management of patients, to improve drugs to deal with the virus. I think we'll see a steady stream of that. Look back to the last 20 year of plague that was the HIV/AIDS episode in American history. That is actually what reduced deaths the most, was work on that. So I do think we will be able to get this under control. And the smaller the problem, the more we can be using testing, tracing, quarantining with improved testing, serious tracing, which can only be done with a small number of people, and isolation. And I think at that point we are back to normal.
There's the question about whether you would recommend one kind of vaccine over another. And none of us are physicians. Does anyone want to comment? One of the things that I feel very comfortable with is how large the studies are and how good the scientific papers are coming out around this, and how public all this data has been. It's made me feel very confident in the process. Does Stu or Carolina, do you want to have anything to say on that?
Before they do, can I just ask, has anybody had a choice?
That is a great question, Arvind, because at this point, it was described to me the other day by somebody, an old quote from Donald Rumsfeld, that you go to war with the army you have, not necessarily the army you want. And at this point, everybody is taking what they can get on campus. Here, we've only had Moderna vaccines so far, and not a whole lot of them, I should add.
Yeah, and when the time comes and we have a choice, is really, the protection will be there. I mean, it is really impressive how these vaccines are reaching 94% efficacy. So, having one dose versus 2 doses, well, that makes it easier. But if it's not available, well, you go with one that has 2 doses, right? The transport, right? So. For one that has to keep a cold chain, that one is going to be not as accessible in places where you don't have this kind of storage, like third world countries and isolated rural communities, for example. They will have to choose the one that can be transported at room temperature, or that doesn't meet these ultra low temperatures. So I think it's going to be more than a choice, a matter of where you are at, what is available to you, and under what conditions can you get the vaccine. So right now, as we said, there's no choice. There's the ones that we have, and that is it. But as the months come, we'll see that more will become improved, and it will be a better scenario.
Well, I think we're going to have to end the panel there. It was a really fascinating discussion on the science and the biology of COVID. And I think we'll turn it back to the Dean for some closing comments.
Great. Yes, thanks to everyone for joining the CCS Lounge. It's co-hosted by CCS and UCSB alumni. Thanks to the audience for all the questions, and we're sorry we couldn't get to them all. We got some great questions. It's been a fascinating discussion and set of responses, so thanks for that. Absolutely special thanks to Professor Belcher for joining us from across the continent. Thanks to our illustrious panelists and to our unflappable staff for making this possible. We look forward to seeing you all at future live events. In the meantime, look for upcoming online events hosted by CCS, UCSB alumni, and others at UC Santa Barbara. And don't forget to stay in touch via social media. So on behalf of all of us here today, we wish you all a good evening. Thank you.